Translation occurs differently between prokaryotes and eukaryotes. In prokaryotes, the RNA polymerase is able to bind directly to the promoter. This process is regulated by the regulator gene, which produces a repressor molecule which then binds to the operator, which prevents translation in prokaryotes. In eukaryotes, the promoter and the enhancer need to be brought together in order for RNA polymerase to bind to the promoter. This is done by attaching transcription factors to the enhancer, which decreases the distance between the promoter and the enhancer. There are many different structures within a DNA molecule, 3 to be exact. First, you have the phosphorus, then the ribose sugar, and finally your bases. Now, the sugars are bounded together through phosphates by covalent bonds, and the complementary bases are bound together by hydrogen bonds. For the complementary bases, A goes to U. In complementary bases, A goes to T and C goes to G. There are 2 differences between DNA and RNA. DNA is made with A, T, C, and G, which makes it a stable molecule. RNA, however, is made by A, C, G, and U, which makes it an unstable molecule.
In replication, DNA would be copied to create two copies of DNA. So the telomeres are used to prevent any damage from occurring to the DNA strand. The topoisomers it relieves the tension of the DNA strand by kind of making it linear and unwinding it. So in the leading strand, DNA polymerase 3 comes through and it binds the complementary strands together, and
The main difference between the lagging strand and the leading strand are Okazaki fragments. Okazaki fragments are basically just fragments of the newly created DNA. These Okazaki fragments, then need to be bound together by DNA ligase.
So in transcription, we have to ensure that the silencer is turned off because it prevents the transcription from occurring. The enhancer activates the what's that say? In order for transcription to occur, we would need to ensure that the silencer is turned off, as the silencer would prevent transcription from occurring, and we would have to activate the enhancer, which would promote the RNA polymerase to come over and bring complementary strands of RNA. So T would go to A, C would go to G, G would to C. However, A would go to U instead of T in this situation.
Once DNA has been fully transcribed, it is sent from the nucleus down to the ribosome. The ribosome is composed of 2 subunits, the large subunit and the small subunit. The large subunit has 3 areas in it: the A site, the P site, and the E site. The A site is where the majority of tRNA goes, besides the first one. The P site is where tRNA binds its amino acids to the previous amino acid to make an amino acid chain. And the E site is where the tRNA exits. The small subunit is where mRNA goes. The first codon of mRNA will always be AUG, which will code for a tRNA. This tRNA will bring the first amino acid in the chain to the AUG at the P site, which will then continue on until there's code to end the amino acid chain.
So in proteins, there are multiple structures. One of them would be the primary structure, in which it is a linear sequence of amino acids that are all joined together by polypeptide bonds. And carbon, nitrogen, and oxygen make up the protein backbone. The secondary structure, however, it creates 2 types. There's the alpha helices, which is a right-handed coil, and then there's the beta sheets, which would be hydrogen bonds stabilized by 2 or more adjacent strands. Over here we have the competitive and non-competitive, in which the competitive would be when other molecules come over and inhibit or promote an enzyme action, but the reaction would still reach the same Vmax. For non-competitive, inhibitors would bind to the bottom of the enzyme, preventing any molecule from entering the reaction, and this would ultimately decrease the Vmax from occurring.